Welcome to the Peak Studio DIA workflow video tutorial. In this video, I will explain how to export a spectral library, how to set up a DIA analysis, and explain the result structure. I'm going to start by showing you how to generate a spectral library from a previous analysis in Peaks. Using a spectral library for DIA data analysis is optional, but can result in overall greater proteome coverage and accuracy. Spectral libraries can be generated both from DDA and DIA data in Peaks, but DDA results are generally used for this purpose. Here I have a sample DDA result. From the export page, check Spectral Library TSV from the database search export. You can also create a spectral library from de novo sequencing results. There are a few options that come with spectral library generation. First, you can choose whether or not to exclude PTMs that do not pass the set confidence thresholds. Unchecking this will include all PTMs found regardless of confidence, although the peptide scores themselves still need to pass the FDR control or -10 log P threshold that is set. You can also choose whether to include the de novo only peptides, which were peptides that were not identified in the database search. A score threshold can be set for the de novo only peptides to be included in the spectral library. The default is 70% average local confidence. Finally, choose Original RT if your LC gradients are going to be the same between the data in the spectral library and the DIA data you wish to search. Choose Map to IRT to map the spectral library data to an indexed retention time. This option is best if you expect your LC gradients to be different between the spectral library and the DIA data to be analyzed. Change the save location if desired. You will need to know for later where you saved it, and click on Export. A .tsv file will be generated. This is the spectral library. As well, a .info file will be created, which contains information about your library. Now, let's set up a DIA search. Click on New Project. Name your project, change the saved location if desired, and click on Add and locate your samples. Click on this button to add each file as a new sample, or use regex to auto-name your samples based on the existing sample name. Next, choose your enzyme, instrument, fragmentation mode, and acquisition type from each drop-down menu. For acquisition, remember to change it from DDA to DIA. Highlight one sample and then copy the whole project so you don't have to change it individually for each sample. You now have the option to choose Submit, which will load the samples without running any analysis. You will then be able to go back later and set up a workflow. For this demo, however, we will continue with the rest of the setup. Click on Next to continue. The workflows in Peak Studio have been reorganized and simplified. Specifically for DIA, the identification and quantification workflows are now listed under one. The two workflow choices are Proteome, which includes the traditional identification and quantification workflows and reports at the protein level, or Peptidome, which reports at the peptide level. For this tutorial, we will focus on the Proteome workflow. The first page you see will be for basic options. Set your error tolerance or keep it at auto detect. If you have more than one sample, you will be given the option to perform match between runs. If you uncheck this box, then match between runs will not be performed at all. If you check it and do not do any further grouping, then match between runs will be performed across all of the samples. You also have the option for group-specific match between runs. Highlight the samples that you wish to group together, and then choose Create a new group for selected samples. You should only perform match between runs between samples that you expect to have the same protein identifications. For example, if you have 3 replicates of a knockout condition and 3 replicates of a control, then you should group the knockout samples together and the control samples together. Next are the spectral library search options. Keep the box checked if you wish to run a spectral library search, and uncheck if you would like to skip the library search and move on to a direct database search. For this tutorial, we will set up the library step. Click on New to configure a new spectral library. Browse to find your spectrallibrary.tsv file and give your library a name. Click on Add Update. You can also select an already configured library in the drop-down menu. The search can be filtered by peptide length, precursor M/Z, or charge. Leaving all boxes unchecked will mean that no filters are applied. There is also the option to select a database for protein inference. This is a great option if you are only performing a spectral library search. If you run a database search, then that database will be automatically used for protein inference for spectral library peptides. The next page contains the database search parameters. Check the database search parameters box to run a database search, or leave it unchecked to skip it and only run a spectral library search. If your sample prep contained a phosphorylation enrichment step, then you can check this phosphorylation enriched data box. This will kick in an algorithm that is specifically designed for phospho-enriched data. You can leave the enzyme as specified by each sample because we already set the enzyme in the project creation step, or you can override that choice here. There are 2 options for digest mode if your sample was enzymatically digested: semi-specific and specific. Specific will only allow digestion strictly following the digest rules of your chosen enzyme. This is a great option to keep a well-controlled search space. Semi-specific allows for one end of the peptide termini to disobey the digestion rules. This option opens up the search a little more and usually results in greater coverage. You can also choose the number of missed cleavages to allow. If none is chosen as an enzyme, then the digest options change. Unspecific means that the database will be digested with no specificity. No digestion means that the database will not be in silico digested at all. This option should only be chosen if your database already consists of peptide sequences and not protein sequences. Click on Set PTM to choose your fixed and variable PTMs. For DIA, we usually recommend limiting the search to around 2 to 4 variable PTMs to keep the search space well controlled. The maximum allowed variable PTMs per peptide can also be set. Next, choose your target database. Click on New to configure a new database. Choose the FASTA format. The options are UniProt, NCBI, and Other. The parsing rules can also be customized. Give your new database a name and click Browse to find your FASTA file. You can choose to validate the FASTA file to ensure there are no issues with the format. Click on Add Update. Be sure that the number of sequences matches up from what you expect with your database. You can also choose to set a contaminant database. The 2 databases will be concatenated and searched together in a single round search. The database search can also be filtered based on peptide length, precursor M/Z, fragment M/Z, or charge. The option to run a spider search is now available for DIA. Under Sequence Variants, check the box to find mutations and sequence variants. The de novo score threshold for the de novo peptides that are included in the search can be changed as well. Find novel peptides is now a checkbox instead of being on its own separate page. Check the box to perform de novo sequencing. The next step is label-free quantification, which is another optional step. Since LFQ will be covered in detail in another video, we will skip it for now. Quality control is also an optional step that will provide statistics about your data based on different attributes that can be selected. It will let you know whether each attribute falls within the set acceptance tolerance from the control. This works best for assessing reproducibility and can be helpful for figuring out why a sample may be deviating from others. The results from QC do not impact your search results at all. It is only meant to provide additional information. You can also search for standard peptides using the QC tool. The final step is the report filter page. For the project level filtering, you can either choose to filter at the protein group level or the peptide level. An FDR or a -10 log P cutoff can be specified. You also have the option to select an additional precursor filter, which will apply FDR or a score cutoff to precursors at the sample level. The number of unique peptides required for a protein to be reported can also be set here. Score cutoffs for sequence variants and novel peptides can also be chosen, as well as the number of common amino acids to the database that are required for de novo tag sharing. The workflow parameters can be saved, so you will not have to re-input any if you wish to analyze other data with these parameters. You can give your analysis a name here, which can be changed later. And you can choose to export your results here. This will export all of the possible results, which may increase analysis time. You can also pick and choose what you want to export later after the analysis is complete. Click on Submit to start the analysis. Now let's take a look at a completed demo project. The result layout is quite comprehensive, and so we won't be covering everything in detail in this video. Instead, this is going to be a general overview of the result structure. For detailed explanations of each figure and all of the contents of the result tabs, please see your user manual found within your Peak Studio installation folder. In the upper left window, you'll see your project name. Underneath will be any analysis that you have run, along with the loaded data. Expand a data file to view the raw data. Peaks will give you an MS1 focused view, an MS2 focused view, an LCMS map, and an LCMSMS map. Expand an analysis to see the refined data and result nodes. Like with the raw data, you can expand the data refine node and view the refined data. This data view will include the pre-processing information as well as result information. For DIA, only the MS1 view and the LCMS view are available for refined data. The interactive LCMS map helps to bridge between the identification results and the raw data for easier result validation. The map can show detected precursors, MSMS spectra, database identifications with and without precursors, sequence variant peptides, and novel peptides. Click on an identification to bring up a supporting fragment ion map. For the DIA search, all results, including spectral library, database search, sequence variants, and novel peptides, are all reported under one result node labeled DB Search. Double-click to open it. The summary page will give you figures and statistics about your results, including retention time calibration information and mass error distribution. The proteins view shows the coverage map of each protein identified with a list of supporting peptides, sequence variants, and novel peptide tags. Supporting peptides will be shown in blue on the coverage map, and novel peptide tags will be shown in gray. The Peptides tab lists each identified peptide with scoring information. The corresponding MS2 spectrum is shown below in a mirror plot against either the library spectrum or the predicted spectrum for the database search. This can be changed to an annotated spectrum instead. By default, the highest scoring spectrum is shown, but other spectra that support this peptide ID can be selected with the breadcrumb feature. The LC-MS maps and profiles are shown at the precursor and fragment ion level as well. The sequence variants and novel peptides tabs also list the peptides identified through these methods. The feature tab shows the identifications from all search steps, including spectral library peptides, database search peptides, novel peptides, and peptides with sequence variants, grouped by feature. The sequence will be listed along with the search engine it was found using. Click on the Export node for all of the export options. Check the options you wish to export, choose a location, and then click on Export. We hope you found this Peak Studio DIA tutorial helpful. Please visit our website at bioinfor.com for more information, answers to frequently asked questions, and other tutorials. Thank you for listening.